lecture, we'll be talking about genetics. In the previous lecture, we talked about variation. So while variation is concerned about the differences among living organisms of the same species, genetics is concerned with the similarity among living organisms of the same species. Have you ever wondered why you so look like your father or like your mother? Have you ever wondered why siblings look so much alike? But the answer to this is genetics. Why are you different from a rat? Genetics. Why are you different from a chimpanzee? Genetics. So genetics is the study of heredity and variation. The study of genetics started by a man called Gregor Mendel, who's called the father of modern genetics, when he studied the pea plants and studied the pea plants Pisa sativum in his backyard, and he noticed gradual color changes and shape changes of these pea plants. And then I was curious and said, why are some pea plants or seeds, why are they round and some are oval? Why are some purple, some yellow? What could be the reason behind this? There was no reasonable explanation to that until the 1900s, when other biologists came up and they furthered the study and they saw that there are driving forces behind genetics, and they call them the genes, and the study of genes is called genetics, and there we are today. For the study of genetics to be possible, a number of organisms are best suited for genetic study, while some are not. The organisms or plants or organisms, generally speaking, that are best suitable for the study of genetics must suit or must fill up or satisfy the following criteria. Criteria number one, they must be small. And the reason they must be small is so they can be kept in large numbers in a laboratory. Number two, the reproductive rate must be very high, so we can study a number of generations even in our own lifetime. Three, they must show conspicuous discontinuous variation. And the reason they must have a conspicuous discontinuous variation is so that we can easily differentiate from one organism to the other, from one organism of the same species to the other. One organism which fits or which satisfies this condition is the African fruit fly. It is an animal. It is called Drosophila melanogaster. The African fruit fly is a very small organism. It has a very high reproductive rate. It reproduces in large numbers at once, and it shows this continuous variation in that the male species have a rounded abdomen and the female species have a pointed abdomen. So we can easily differentiate between the male and the female. So what are those factors we need to familiarize with? What are those terminologies we need to familiarize ourselves with for us to understand genetics? Number one is the gene. The gene is called an hereditary unit. A gene is a sequence of DNA that is found within the nucleus of a cell, and these genes are bodies which code for certain traits or characters within the organism. A trait or a character is the distinguishing feature of an organism. Now, genes are found on specific location of chromosomes. The location on which these genes are found is called a locus. The chromosomes are hereditary carriers because they carry the genes, which are the hereditary units. So chromosomes are hereditary carriers. They're found within the nucleus of a cell. A cross is a match made between two individuals to produce offsprings. The offsprings produced are in a generation and are called a filial generation. A genotype is the genetic makeup of an individual. It means what is genetic makeup, what the gene expression on an individual, the gene contained in an individual is genotype. The phenotype is the physical makeup of an individual, the expression of the genes. For example, height is a phenotypic character, skin color is a phenotypic character because we can see them, they are the expressions of the genes of that individual is what we can see. The physical makeup of an individual is called its phenotype. There is a condition where the genetic makeup of an individual changes suddenly. That procedure is called, not process, is called mutation. Mutation is a sudden change. The law of segregation of genes. The law of segregation of genes states that during gamete formation, genes separate or segregate into the respective gametes, where each gamete carries only one copy of the gene and is transmitted to the offspring in that manner. What that means is, for example, the male gamete is XY and the female chromosome is XX. It means that the male gamete will carry one of the two chromosomes. So some sperm cells will carry chromosome X and some other sperm cells will carry chromosome Y. So chromosome XY segregated, that means the split, and each copy of the gene are registered on the gamete. X is a gene, Y is a gene, XY is an allele. XY is an allele is a pair of genes which controls contrasting characters. And when XY splits, you have X going to the first gamete and Y to the second gamete. So the law of segregation of genes says that during gamete formation and during fertilization, the gametes fertilizing each other have only one copy of the pair of genes. So the X cell has one X, another X cell has another X. One sperm cell has X, another sperm cell has Y. So it now depends on which sperm cell fertilizes the egg first. If the X sperm cell fertilizes the egg first, you form a female, which is XX. If the Y sperm cell fertilizes the egg first, you form a male, which is XY. So the law of segregation of genes, which is Mendelian's first law, explains to us that genes actually segregate, or alleles actually segregate, and they move into the gametes singly. The second law is law of independent assortment of genes. Independent assortment of genes states that genes are inherited independently of others. So if I'm to inherit the gene for tallness, for example, the gene for tallness has nothing to do with the gene for skin color. The gene for skin color has nothing to do with the gene of my blood group. So all genes are inherited independently of others. So gene 1 is inherited independent of gene 2. Gene 2 is inherited independent of gene 3, and it goes that way. And the Mendelian law of dominance states that in an individual with dominant and recessive traits, with contrasting traits, the dominant traits usually gets expressed over the contrasting traits. For example, if I have the allele for tallness to be big T, small t. Big T, small t, the big T in this pair will always be dominant over the small t, and this individual will be tall, all because it has a big T. So we have the dominant traits always expressed over the recessive traits. There could be a cross between, for example, a dominant tall man, TT, and a dominant short woman, small t, small t. Now, a dominant tall man means zygous or homogeneous tall man. Homogeneous or homozygous tall man means the tallness is homozygous, that's big T, big T. And homozygous dwarf woman, that's small t, small t. When you have a cross, one big T crosses with the small t, this other crosses with the small, this big T with the small, this big T with the small, and it gives you all tall offsprings. You're going to be that it's all tall offspring because the tallness, which is dominant, will always be expressed over shortness, which is recessive. Alright? And the phenotypic ratio will be altered, while the genotype contains big T and small t. I hope we understand. The same. Okay, let's take this question. An hemophilic man marries a normal woman. Full stop. What percentage of their offspring will suffer hemophilia? Now, the man is XY. Sex-linked traits are always registered on the X chromosome because both males and females are, well, it depends. It depends, but both males and females have X chromosomes. So you have transmittable traits, transmittable characters registered on the X chromosomes. So if we have sickle cell on the X chromosome, for example, and normal cell on the Y, and then the X here for the woman is normal, by the time you have a cross, the cross gives you two abnormal females, two females who are carriers. A carrier is an individual which has both contrasting character. For example, big T, small t, this is a carrier, is a tall man with a carrier for dwarfness. Okay? Say this is AS, which is a genotype. The genotype for this individual is, if the individual is AA, is homozygous normal. SS, homozygous abnormal. AS is a carrier because the individual is normal but carries a copy of abnormal traits. So AA is an homozygous normal individual, SS is an homozygous abnormal, while AS is but a carrier. Okay, so Mendelian's law gives us the proper knowledge to crossing of parents to give offspring. Is it advisable for an AS man to marry an AS woman? The answer is no. Because if an AS man marries an AS woman, they will give birth to AA, possibility AA, AS, and SS. So there is a 25% possibility that they birth an SS child, which we don't want to happen. So the SS will be avoided, and the AS man, AS woman will be advised not to marry. Can an AA individual marry an AS? Yes. Because you have AAAS, you have AAAS. All are normal, although two are homozygous normal, two are carriers. And the carriers are the AS. So in this kind of a case, you have 50% homozygous normal child, 50% carriers.